Diagnostic and detection method. There are many types of applications used in diagnostic and detection method of immunology, such as antibody generation, immunoprecipitation, agglutination reactions, antibody assays based on molecule binding to solid phase supports, affinity of antigen-antibody interactions, microscopic visualization of cells and subcellular structures, immunofluorescence-based imaging techniques, and lastly flow cytometry. In this video, we will be focusing on antibody assays based on molecule binding to solid phase supports. The most commonly used method is ELISA. ELISA is used to measure antibodies, antigens, proteins, and glycoproteins. They are generally carried out in 96-well microtiter plates, and these plates are special absorbance plates, for example, NUNC immunoplates, to ensure the antibody or antigen sticks to the surface. Each ELISA measures a specific antigen, hence there are kits available for a variety of antigens. How ELISA works? Firstly, ELISA plate is coated with captured antibody, which then the excess of unbound antibodies will be washed away from the plate. Next, the samples such as urine, serum, or cell supernatant will be added. Any antigen found in the sample will bind to the captured antibody, which is already coating the plate. Again, any excess sample will be washed away from the plate. Next, detection antibody will be added. This antibody is labeled with an enzyme, usually horseradish peroxidase or alkyne phosphatase. Detection antibody binds to any target antigen that is already bound to the plate. Finally, a substrate is added to the plate. ELISA assays are usually chromogenic, using a reaction that converts the substrate, as example TMB or ABTS, into a colored product which can be measured using a plate reader. There are three types of ELISA formats, which are direct assay, indirect assay, and captured assay, known as sandwich. There are three types of advantages, which are quick because only one antibody and fewer steps are required. Secondly, they are versatile because many primary antibodies can be made in one species and the same labeled secondary antibody can be used for detection. Thirdly, the sensitivity is increased because each primary antibody contains several epitopes that can be bound by the labeled secondary antibody, allowing for signal amplification. The disadvantages of ELISA are time-consuming and expensive. Secondly, there are minimal signal amplification, and there is no flexibility in choice of primary antibody labeled from one experiment to another. Second is immunoblot, Western blot. The HIV immunoblot is used for confirmation of HIV infection. The steps involved in HIV immunoblot are: the blood sample taken from patient is loaded and they are separated on STS page. The viral proteins will then be transferred into a nitrocellulose membrane, and the membrane will be blocked with neutral protein such as BSA or milk casing. The next step will be incubating the membrane with primary antibody specific to the target protein. Next, it will be incubated with HRP labeled secondary antibody which is specific to the primary antibody. And finally, it will be incubated with chemiluminescent HRP substrate and exposed to the film. Finally is the rapid test. Rapid test is used to detect HIV antibodies in the blood, mostly HIV-1 and HIV-2 antibodies. They work by capturing antibodies or antigen on a solid surface and then attaching molecules to them that allow detection by the naked eye. There are two types of HIV rapid tests which are present. They are immunoconcentration and immunochromatography. Immunoconcentration involves dot test, whereas immunochromatography involves line test. In immunoconcentration, flow-through devices are used, which are usually cartridges with HIV antigen attached to a membrane. HIV antibody links to the HIV peptide antigens, forming the color spot. To interpreting the results, H negative produces all colorless dots except for the control. Next, for HIV positive, which involves dual reactions, will show red in all the three dots. HIV-1 positive, red color will be present at the control dot and the HIV-1 dot. And finally, if the result shows HIV-2 positive, red color will be present at the control and the HIV-2 dot. Immunochromatography involves line test which uses lateral flow devices. HIV negative shows non-reactive result where only control area shows the line. Next, HIV positive will show reactive result in both line of the control and the test area. If there is no any line showing, it shows that the result is invalid and requires test repetition. The other application of immunochromatography are HIV half triline rapid test, which the test is based on immunochromatography. Secondly is the INSTI HIV-1, HIV-2 antibody test. It involves a single-use, rapid, in vitro qualitative immunoassay for the detection of antibodies to HIV-1 and HIV-2 in human. Thank you for watching and hope you all enjoy the video. Till then, see you all in the next video.